Okay. Right.
All right. Everyone can take a deep breath after being scared that you missed the abstract deadline. So today I'll be introducing Dr. Cisneros. She received her Bachelor of Science in Biology from the University of Texas at Brownsville. She then received her PhD in Cellular Neuroimmune Pharmacology from the University of North Texas Health Science Center. And she did a postdoc at UTMB and is currently an assistant professor of pathology there. And today she'll be telling us a little bit about her research journey and her research interests around neuroinflammation.
So this is one of my favorite talks to give, but this is kind of unique because I get to sprinkle in my research. So you guys are going to hear a lot about my science and a lot about my background. So I'm also very transparent. So don't get scared. So I grew up in Harlingen. So that's the tip of Texas, near Mexico. My mother was an LVN, but when I was growing up, she wasn't practicing. She owned an antique shop. So she's very creative, so I think I got a lot of creativity from her. My dad moved to Harlingen or actually Raymondville, when he was 12 from Matamoros. And he graduated from high school when he was 21 because of the English and the language barrier. So my dad never taught me, nor my sister, how to speak Spanish because he always associated Spanish with poverty. So I have picked up a lot on Spanish. My husband is very fluent, and I remember being around his mother, and she would just like try and communicate with me, and it was very hard. But I have picked up on Spanish now, and I'm able to communicate a little bit better. But I've always had an interest in science ever since I was little. And one of the things that really sparked my interest was I had a fourth grade teacher. Her name was Miss Vincent. And she used to come to class with like her python around her neck, and she dissected a pregnant shark in front of us. And in fourth grade was actually the first time I had to do a science fair. So me and my best friend got together and we did a volcano because that's like super cool. And so we had it erupt and everything. And then when our teacher came up to us and asked me what our hypothesis was, I was stumped. I was like, what the heck is a hypothesis? I got a B and I was pissed because I was a straight A student and I was a little bit discouraged. But then when I was in sixth grade, I had to come up with a new science fair project. So I tested various types of conservation, and I did this with like first and second graders. Now that I think about it, I probably needed IRB approval, but I was a sixth grader, and I was there and I had a hypothesis and I did really great and I loved it. So in sixth grade was also the time that I first watched the movie Outbreak. Does anybody know what Outbreak is? Oh my God, I love that movie. When my dad would tell me the blockbuster was the movie, I always rented, took it home. I wanted to work with Ebola. I wanted to wear the big yellow suit. I wanted to go to Africa. That's what I wanted to do. But I didn't know how to do it. Where I grew up, nobody really knew what scientists were. I mean, it was in the 80s. And it was unique for a little girl to always say, I want to do this. People were like, what's Ebola? They thought I was crazy. Well, in high school, I had another science fair project and I made it very microbiology specific. So I tested different bacteria on different surfaces and like different agents that would kill that bacteria. And I actually made it to state. Still, nobody in my high school could really help me decide on the path to get to be a scientist. I did end up graduating from high school with like 8 science credits. Any elective I could take, I took a science class. I was always in those rooms during lunch and, I was a nerd and I just love science. So then after high school, when I graduated from high school, I applied to several colleges. I got into Cornell, Baylor, UT Austin, but because I was dating somebody, I stayed local and I went to school 40 miles from my home. Edinburgh, Texas, UTPA. Well, my parents were extremely conservative when I was growing up. I always had a curfew. I never got to go out. So when I got to college, they paid for my apartment, I had a credit card, they bought my groceries. But did I go to class? No. I was at the raves. We lived so close to Mexico, I would drive to Mexico, we were partying all night. I never made a class. I failed my whole first semester. I got involved in recreational drugs. I failed my second semester, got kicked out of college, started community college, lied to my parents so my dad would keep paying my bills. And I kind of got really messy with substances of abuse. I met my husband. We both struggled a lot with substances of abuse. And I say this because it kind of formed where my research has gone now. And so we got married because we wanted to move to Dallas, and my parents were like, "You're not moving unless you're married." So we got married and we moved to Dallas, and a few months later, I got pregnant. So I stopped using drugs, and I ended up, having my baby. And then when we got, after about a year later, after our son was about a year, I decided that I wanted to go back to school. So the only place that I could really do that was in the Valley, where my parents could help me take care of my child. So we moved back to the Valley, and I went to school at UTB. So University of Texas at Brownsville, now UTRGV, and ironically now is joined forces with UTPA. So now they're one big university down in South Texas. But I got to University of Texas at Brownsville in 2005. And when I got there, I became a fellow for the Minority Biomedical Research Support Research Initiative for Scientific Enhancement. So this was like the first time that I actually had a chance to experience bench work. And so I was a fellow for this from 2005 to 2007. The first lab that I worked for was for Masako Isokawa, and she was a cannabinoid receptor biologist, electrophysiology, and she was studying how nicotine affected cannabinoid receptors. So that was kind of my first experience with some sort of substances of abuse in research. So then a few months after that, I joined Dr. Michael Laker's lab. And Dr. Michael Laker was actually the director for the MBS RISE program at the time. And my research with him was with Trichomonas vaginalis. So that's a parasite and an STD. And so that was my first time I actually had an opportunity to work with sexually transmitted diseases. So already during my first kind of research experience, I had an opportunity to work with some sort of substances of abuse and with STDs. And I say that because now my research is really concentrating on substance use disorder and HIV-associated disorders. So then after I graduated in 2007, I applied to some graduate schools, but I didn't get accepted into anywhere. So I decided that I would take a year, and I applied at University of Texas Southwestern Medical Center. And I became a lab manager and a research associate for Dr. Chandrasekhar Pasari. And this was immunology research. So this was the first time I had ever really been introduced to immunology other than like my undergrad courses. And I vowed I would never do immunology again. It is so hard. So kudos to any of you that work with like T cells and B cells, because I still don't get it. Like it's complicated. I was there for a year, and then I got pregnant again, and I decided that I would take that year off when I got pregnant. So during that year, I started applying to graduate schools again. I actually applied to a marine biology school in South Carolina. Thank God I didn't get in because I'm scared of the ocean. Like, I don't know, but I ended up getting accepted to the University of North Texas Health Science Center in Fort Worth. Orientation for graduate school was on May 22nd, 2009. And that's actually the day that my daughter turned 1 year old. So I'm sitting in orientation. I'm the oldest graduate student there, or at least I think I am. I have probably the only one with children, and I'm nervous. I'm sitting in this room with like all these kids and I was excited because I became a fellow for the Minority Biomedical Research Support MORE program. So this is kind of piggybacking on the MBRS RISE program that I was involved in at UTB. And I really think that the reason that I was part of that program is how I got into graduate school.
because, you know, that CV and that track record of accomplishments. So while I was there, that first semester, I failed. I am very detail oriented, so I concentrated a lot on trying to memorize all those details instead of the overall picture. And so I was on probation after my first year of graduate school. Then my second semester, you know, I got better. I understood a little bit more about the science and what they were expecting of me. And then I had to do a rotation. So I was looking for the only lab on campus that had virology research, and that was with Dr. Anusha Gorpati. She was an amazing mentor. She taught us how to write publications and manuscripts, and grants. She had 3 R01s when I was in her lab. She was the highest funded PI at UNT Health Science Center while I was there. And she had kids, you know, like she understood the sacrifices I had to make to be there. But I do remember that the first time that she gave me an evaluation, she gave me unsatisfactory. So I lived an hour away from graduate school. And after she gave me that unsatisfactory, we sat down and she was like, I think you need to move closer. So about a month later, we found a house that was 2 blocks from the university. My kids' elementary's crosswalk was across the street from our house, and my daughter's daycare was in between, the block in between. So after that, I was dedicated. And so the research that I ended up getting involved in, she gave me her R01. She was a multi-PI and was responsible for 1 AIM. So she asked me to read the AIM, and the first thing that she charged me with was writing a review. So the review that I ended up writing on was HIV, methamphetamine, and astrocyte glutamate regulation, combined excitotoxic implications for neuroAIDS. And I was able to put 1 piece of data into that into that review. And so the data that I was able to share was that we looked at expression of excitatory amino acid transporter 2, which is the glutamate transporter responsible for 90% of glutamate uptake, and it's primarily expressed in astrocytes. And so we found that inflammation, meth, and HIV all lead to downregulation of E2, and glutamate excitotoxicity is actually one of the things that leads to HIV-associated neurocognitive disorders. So the review was published, and then she asked me to kind of develop my dissertation project from, you know, the literature that I went through. So the biggest gap that we decided to address was the mechanism that methamphetamine used in astrocytes to signal and induce E2 downregulation. So going through the literature, we identified traceamine-associated receptor 1, which is a G protein coupled receptor, and it was shown that it can be activated by methamphetamine in neurons, and it increases cyclic AMP and protein kinase A activity. And to connect it to E2 transcription is also controlled by protein kinase A activity. So one of my other interests during this time was looking at hypothermia, because methamphetamine abuse and HIV both can lead to brain hyperthermia. So I kind of joined that into my dissertation work. And I started by looking at primary human astrocytes. So in this lab, we used primary human fetal astrocytes, and we treated them at 39 degrees. And you can see that the phenotype for the astrocytes was already activated just with a 2-degree increase in temperature. So then I submitted at F31. And I titled it Meth HIV Regulation of Astrocyte Responses, TAR1 and Hypothermia. And I'm not going to read the hypothesis, but this was the hypothesis for the F31. And my program officer was Dr. Albert Avila. So the one thing that I can encourage everybody, and I always tell everybody, you make friends with your program officer. So I met Albert a year before when I was planning to submit an F31 at a conference. And we sat down and we talked. And so he could put a face to the name when the application came in. And we talked right after it was submitted, and, you know, he gave me all the news of the study section. And even to this day, we're still in contact. He's the NIDA diversity officer now, chief officer, but he was a really big asset when it came to funding of my F31. So I encourage everybody to always keep in contact with your program officers. So what I did as a pre-doc was we looked at TAR1 in these astrocytes, and we found that HIV plus methamphetamine increased TAR1 expression. And we saw that HIV, meth, and combined conditions increase cyclic AMP. We also saw that astrocytes do express TAR1 pretty much everywhere in the cells. So we saw it in the nucleus, we saw it in the cytoplasm. Ironically, not on the membrane, and being a G protein coupled receptor, you'd expect it to be on the membrane. We also saw that meth and the TAR1 agonist beta-phenylethylamine all could increase intracellular levels of cyclic AMP. And this graph right here was individual biological donors. So you can see the average EC50 for each of those agonists. Then we downregulated TAR1 in astrocytes. And this is just showing that it was efficiently downregulated and that when we downregulated TAR1, meth and beta-phenylethylamine no longer induced cyclic AMP. So those increases in cyclic AMP in astrocytes were through TAR1. But one of the most interesting parts of our data was that when we downregulated TAR1 in astrocytes, meth-induced E2 downregulation was lost, and astrocytes' ability to clear glutamate was increased. And when we overexpressed TAR1, E2 downregulation was increased, which sounds counterintuitive, but and glutamate clearance was significantly reduced. This implies that TAR1 is somehow involved in glutamate clearance and E2 regulation. So as a pre-doc, part of what you're doing is you're growing your CV. So I was an introvert who had to become an extrovert. During my first talk, my PI also had told me after, my presentation, she was like, why are you flirting with the audience? You're always flipping your hair and like you're acting coy is the word that she told me. And so I take constructive criticism really well. It didn't hurt my feelings. But I ended up going and taking speech classes and joining Toastmasters and taking her advice and using it to become better at presenting. And so I just say that because everybody has to take constructive criticism, right? So to become an extrovert as a pre-doc, I became secretary for the Black Graduate Student Association. I was vice president and president of the Graduate Student Association, and I applied to everything. I didn't let an application pass me by. It helped me learn how to write applications. It helped me learn how to take constructive criticism, and it helped me learn how to fail because I didn't get funded for everything. But during that application process, I did get funded as an associate fellow for an NIAT 32. I was a NIDA Diversity Scholars Network Fellow. I was an NIH Future Leaders Fellow, and I was an NINDS Brains Fellow. So when you apply, you can get, funded, but if you don't apply, you're never going to get anything. So I defended with distinction in April 2015, and I got like Best Graduate Student Educational Award, which for me was ironic because I failed my first semester of graduate school, but they saw potential. I ended up doing a postdoc with Anuja for a year, and everything kind of came full circle when I was invited to be a keynote speaker for the MBRS RISE program at UTB, where I got to come and talk to the new students. And it was just really nice to be recognized and to show the success that the program has the value of the program in helping you achieve your goals. But my pre-doc mentor, who is now my postdoc mentor, told me that she wanted me to move on. And not because she didn't want me there anymore, but professionally, she told me that it's not good to stay at the institution that you got your doctoral degree at, and that it looks better professionally if you move to a different institution. So I started sending out my CV. But during that time as a postdoc, I submitted a paper to JBC that it didn't get published while I was there. And we actually didn't finish the paper until the pandemic when I was at home and I had all the data in front of me. So just to kind of summarize what that last paper looked like was we saw that HIV and IL-1 beta increased TAR1 and decreased E2, and that meth signaled through TAR1 to increase both cyclic AMP and intracellular calcium. And that both of these signaling pathways that led to both PKA and CAMK2 activity. And when we saw increases in PK and CAMK2, they led to phosphorylation of CREB, both co-activator and co-repressor forms of CREB. And we could manipulate these pathways and we could either activate or repress E2 transcriptional activity by using Cy-PKA or PKA inhibitors, or the same with Cy-CAMK2. So then this was published. Well, Dr. Gopati had a lab manager. Her name was Kathleen Borgman, and she ended up getting a PhD after I left, and she took over my dissertation project and also took over all of the R01s that Anuja had, because Anuja moved on. And now Kathleen is a NIDA program officer. And we talked not that long ago, and I asked her, because now nobody's working on the TAR1 project. And I asked her if it was okay if I submitted an R01 on the TAR1 to NIDA. And she was really enthusiastic about it. Obviously, she can't be my program officer, but she was very enthusiastic about it. So about a month ago, I submitted an R01 on my predoctoral work, and it was targeting astrocyte-induced glutamate toxicity in methane HIV1. So fingers crossed, it at least gets discussed. So when I sent my CV out, one of the places that I sent my CV out was to University of Texas Medical Branch in Galveston, to the Center for Addiction Research. And I didn't hear anything back. And then 3 months after I sent my CV, I get a phone call from Dr. Cunningham. And she was like, I saw your CV. We want to bring you over here as a postdoc. I'm in the Grand Canyon. I'll call you back, like next week. And it was like a whirlwind. I was like, okay. So then when I ended up talking with her, she told me that UTMB had started this new initiative called the Neuroinfectious Disease Initiative. And UTMB is very strong in infectious diseases and in neurosciences. So, but there was no connection between the two. So she needed somebody with a background in both neuroscience and infectious disease to come and kind of be a liaison. So she brought me on as a T32 fellow. And I wanted to do something that was not methamphetamine and HIV. So cocaine is one of the most well-studied substances of abuse there at UTMB. So I decided, okay, I'm gonna do cocaine research. And any virus, I could have really picked any virus I wanted to work with, but it couldn't have been a lethal virus, because why am I gonna study substances of abuse with Ebola, right? There's really no connection there. So I had to think outside of the box, and I thought about Zika. So at this time, Zika was really sexy. Everybody wanted to study it. In Miami-Dade, the highest Zika cases were happening. Cocaine abuse also was the highest in the Miami-Dade County, and Zika is neurotropic. So that was kind of how I connected the dots. I was going to study cocaine and Zika. Everybody thought I was crazy, but I was like, I'm going to do it. So I wanted to start by working with my 2 postdoc mentors that we identified. So I ended up working with Bobo Paisler, who's an arbovirus expert, and he also was starting to reach into the neurovirology field. So now he works with Lassa, and he's studying hearing loss in Lassa. And my other co-mentor was Kelly Dineley, so my infectious disease mentor and my neuroscience mentor. And Kelly Dineley is a behavioral expert and a rodent behavioral expert and an Alzheimer's disease expert. So what was really nice is that they allowed me to do whatever I wanted. They allowed me to do the cocaine and Zika project, but they also had another project where they were looking at Venezuelan equine encephalitis virus and how it exacerbated Alzheimer's disease pathogenesis. So I kind of dabbled in that and did a little bit of behavioral research in that too while I was a postdoc. So what I wanted to do though with the cocaine and Zika project was I wanted to give mice that were infected with Zika cocaine and then look at some of the neuroinflammatory outcomes. Well, it turns out that the mouse model was an interferon receptor knockout mouse. And when I went through the literature, there just was nothing about how cocaine was impacting the interferon signaling pathway. So the first thing I had to do was look at that. And so in astrocytes, I treated them with increasing doses of cocaine, and we saw trends in IL-6 and IL-8. But what was really interesting is that at lower doses of cocaine, we saw an increase in interferon beta and ISG, interferon stimulating gene 15. So that was really ironic because type 1 interferons usually only increase through genomic material of, pathogens. And obviously cocaine doesn't have any of that. So then to kind of further investigate, we found that cocaine also increases DNA fragmentation and increases intracellular levels of ROS. And we can reduce cocaine-induced increases of those outcomes by using glial modulators and antioxidants, propentylphylline and pioglitazone. And we also found that cocaine modulates HIV and Zika-induced immune and interferon responses. So IL-6, we saw that cocaine increased, but both interferon beta and IL-8, we saw that cocaine caused a decrease. So it might have been a saturated response or something. We still are not sure why it decreased those outcomes. But we did see that propentophylline and pioglitazone could reduce cocaine-induced interferon responses and HIV responses. So we kind of summarized all of this in a manuscript as my first postdoc manuscript, where we showed that cocaine evokes a profile of oxidative stress and impacts innate antiviral response pathways in astrocytes. So the next step was, what is the mechanism that cocaine is using to induce this type 1 interferon response? So I identified mitochondrial antiviral signaling protein, which is a pattern recognition receptor, and it's actually activated by self-DNA, including double-stranded DNA, mitochondrial or nuclear DNA that's released into the cytoplasm. I wrote an F32 to NIDA on this subject, and I focused my F32 on Zika, and it was submitted, it was discussed, and it was scored, but I didn't think it was going to get funded. And my program officer, Woody, emailed me and he was like, can we talk? So we talked on the phone and he told me, if you switch from Zika to HIV, your grant's going to get funded. So I did what he said and I got my F32. I still did Zika, but I included HIV. And so some of the stuff that came out of that was we looked at MAVS aggregation with cocaine. We found increased aggregation in the mitochondria. And one of the hypotheses was that cocaine was leading to changes in mitochondrial toxicity, and that's what led to MAVS aggregation and activation. And so we looked at MPTP opening in the mitochondria, and we found that cocaine causes the pores to open. And we looked at MTT and LDH, which are, metabolic outcomes. So about this time, I think it was late 2018, and I got invited to UCSD to go interview. I didn't tell anybody it was for a research position. It wasn't for an assistant professor position, but everybody assumed it was. So I'm in San Diego and I'm giving a talk, and then Bobo, my postdoc mentor, calls me and he was like, don't take the position. I got you an assistant professor position at UTB. And I was like, okay. So then when I got back, I was promoted to assistant professor in April 2019. And this was very exciting because now I got to explore. I had a lot of interest and I got to expand my research and explore all kinds of things with my startup. And this is kind of this is my lab logo, but it summarizes everything that I do. So astrocytes, making up the biohazard sign because we do infectious disease research, substances of abuse, and then the brain, because we look at how neurodegeneration is exacerbated during some of these processes. So the first thing that we did while I was an assistant professor was we did self-administration of fentanyl in rats, and we looked at neuroinflammatory markers. So we isolated different brain regions, and we had lifetime intake of fentanyl in these rats for low and high intake. They self-administered for 14 days. None of them died because, is anybody familiar with self-administration? They have a lever, so they're only allowed to get so much fentanyl for a certain amount of time. They can push the lever as much as they want, but if we allowed the fentanyl to be given every time they push the lever, they would overdose. So there's a cutoff for them. So we are able to calculate how many times they push the lever. And so self-administration is actually really important when we study substances of abuse because it allows the animal to develop the neurocircuitry of addiction versus exogenously giving the animal drug, which doesn't let them develop that neurocircuitry. So we did this, we looked at different brain regions and we just looked at different inflammatory profiles. We found that some of them were increased or decreased when we look at interferon, depending on the brain region. And we also correlated that to MAVS. So we saw some positive correlations between MAVS expression and some of these pro-inflammatory and type 1 interferon responses, specifically within the hippocampus and prefrontal cortex. We also looked at another pattern recognition receptor called STING, but for time, I'm not showing all the data, obviously. And so during that time after this manuscript was published, an RFA came out with NIDA. And is everybody familiar with RFAs? So those are specific calls from the different grant institutes, and they have a very specific set of money for a specific project. So for this RFA, it was inflammasomes and HIV and substance use disorder. So we gathered some preliminary data, and I decided I would submit a grant to this RFA because your chances of getting funded are much higher than when you submit to a parent announcement. So I submitted aiming at the inflammasome. AIM-2 is an inflammasome involved in inflammation, impact of MAV signaling and cocaine and HIV-induced neuroinflammation. And so this was my first R01. I got it 2 years after becoming an assistant professor. It expires 2026. So my graduate student has started working on the effects of cocaine on MAVs and inflammation. So she is actually looking at activation of MAVs by cocaine upstream. So I mentioned that cocaine is increasing levels of double-stranded DNA, and that could be the mechanism that's activating MAVs through self-DNA. So that's what her interest in her project is concentrating on. So she is first looking at how cocaine is increasing levels of DNA in the cell. So through collaborations with Dr. Szczęsny, we looked at mitochondrial DNA copies, integrity, and nuclear DNA. We didn't find any significance, but we do see some trends happening here. So this is implying that if we maybe increase the number of samples, we might be able to find significance in the levels of mitochondrial DNA and nuclear DNA present in the cell, which would imply that this may be the mechanism through MAVs activation. She then looked upstream of MAVs. So there's MDA5, I'm sorry, is a pattern recognition receptor that needs to be activated in order for MAVs to be aggregated. So she just looked at MDA5 levels with increasing cocaine concentrations. And again, we didn't find significance, but we do see some trends. And then we looked again at MAVs aggregation and levels. So we first looked at MAVs protein levels with cocaine, and MAVs can either be cleaved or aggregated. So we looked at both cleavage of MAVs and we looked at aggregation of MAVs. And we found that there is increased aggregation of MAVs following cocaine exposure. And because MAVs is mostly located to the mitochondria, we did mitochondrial and cytoplasmic extraction. Then we looked at expression of MAVs by confocal microscopy, and you can see that increasing levels of cocaine do cause aggregation of MAVs in astrocytes. So when we downregulate MAVs in these astrocytes, we also see that cocaine-reduced levels of IL-10, which is anti-inflammatory, is lost. And cocaine increased levels of interferon beta. It's not completely lost, but it's partially lost. So MAVs is probably not responsible for the entire interferon beta pathway that's activated by cocaine, but at least partially responsible. So my research scientist, Shanita, is now looking at MAVs regulation and downstream of MAVs with cocaine. So she has an overexpression model. And she, again, just treated this overexpression model with cocaine, and we see an increase in MAVs aggregation. And then she looked at all of these different proteins that bind MAVs and induce pro-inflammatory and type 1 interferon signaling. And she found that in this overexpression model with cocaine, we do see an increase in all of these markers when we overexpress MAVs in astrocytes. So another part of this hypothesis of MAVs activation is changes in mitochondria. So Nicole was actually my very, very first employee. She is doing transmission electron microscopy, and she's looking at astrocytes, mitochondria following cocaine exposure. And you can see that 24 hours of cocaine or 48 hours of cocaine, that the mitochondria do look damaged compared to our untreated controls. So my postdoc crit is looking at differences in fission and fusion proteins for mitochondria. And we can also see that increasing doses of cocaine are leading to increased expression of some of these proteins, which implies that there is some mitochondrial dynamics happening during cocaine exposure. He also looked at phosphorylation of DERP1, and he saw increases in DERP1 with cocaine. So now he is working on looking at levels of ATP, mitochondrial reactive oxygen species, and mitochondrial membrane permeability. This one was, I'm so sorry, it's not labeled. It's MPTP opening, so the transition, mitochondrial pore transition opening. And so you can see again that cocaine is impacting all of these outcomes of mitochondrial health. So we do suspect that cocaine is leading to MABS activation through mitochondrial toxicity and through increases of double-stranded DNA. So all of this data is being prepared for publication right now. And hopefully it'll get submitted soon. But one of the new areas that we're going into is it turns out that MAVs, there's 12 SNPs within the coding region of MAVs. And what's really interesting is that individuals who are homozygous for S409F and Q198K actually have lower HIV viral loads and delayed increases in viral loads. And they can initiate and have differences in CD4 T cell populations. Again, immunity, right? But I have to go there. I don't like it, but I do have a graduate student who's a medical doctor. So this is her project and she's kind of initiated it. So she is looking at these MAV single nucleotide polymorphisms in HIV. So this is really complicated, but really all I want you to concentrate on is that we have our MAVs major genotypes and our MAVs minor genotypes, and some of the outcomes are related to Th1 responses, and the mechanism responsible for this is PLK1. Well, we don't know from the literature what substances of abuse, how they influence PLK1 activity. So that's where Mauda is starting. She's looking at whether cocaine and fentanyl regulate PLK1 in peripheral blood mononuclear cells. So she is isolating PBMCs from healthy donors, and then she is first genotyping them to see if we can find any with a minor or major genotype. Right now, she hasn't done many, but this is what she's found. We haven't found anything with a minor, but only 2 to 5% of the population actually have the minor MAVS genotype. And then what she's doing with these PBMCs is she's looking at metabolic activity, and she's doing Western blot, qPCR, and ELISAs after she's exposing these PBMCs to cocaine or fentanyl. So right now, this is the data that she's collected where she's looked at MTT activity of these PBMCs following cocaine or fentanyl exposure. We also have some preliminary data that suggests that fentanyl is leading to differentiation of CD4s into Th1 cells, and that's still really preliminary. But this project is really kind of expanding, and we are going to be introducing humanized mice with some of these PBMCs that are expressing MAVs or major or minor genotype, and then we're going to be doing behavior infecting with HIV. It's like this really big project that we're trying to get funded also. But this isn't all of the work that I do. This is the end of the data I'm going to show you. But right now, a lot of the work that we've been concentrating on is substance use disorder during HIV, infectious diseases. But a lot of the work that we've published recently is on Venezuelan equine encephalitis virus and how it induces Alzheimer's disease pathogenesis through the tryptophan kynurenine pathway. And this is just kind of a list of other R01s that I've submitted to give you an idea of what my research agenda is involved in. So I had an R56 where we were looking at targeting IDO and SARS-CoV-2 induced Alzheimer's disease progression. That just ended in August, but we did get 2 papers out of that with ViiV, and we did a longitudinal study where we infected mice with SARS-CoV-2. We did behavior every 3 months, and we collected biochemistry or tissue for biochemistry at every 3 months. So right now we're processing the samples, analyzing the data. It's a huge project. I got an NIH loan repayment program grant. Is anybody familiar with that? You guys have got to apply for this if you have student loans. So the NIH will pay your student loans back. So it's called NIH LRP. So look it up, put it down. I just got funded for this, so they're paying back my student loans. And the title of that was Delineating Neuroinflammatory Mechanisms Resulting in Neurological Disorders and Sequelae Triggered by Neuroinfectious Pathogens. So NINDS is the one that's funding my NIH LRP. I submitted an R01 on ViiV and alpha-7 nicotinic acetylcholine receptor molecular mimicry, and also post-neurological sequelae of SARS-CoV-2 and Alzheimer's disease progression. This went to study section. It was scored, but it's probably not going to get funded because it was outside of the scoring range. But I think that the program officer is going to support another R56 for this grant. So this is my family. My kids have put up with me. My husband has put up with me through all of this. And I'm also just really passionate about mentoring. This is my lab family. You spend just as much time with them as you do with your real family, if not more. And so this is just all of them. Shanita's my research scientist. She's been with me for almost 3 years, and now she's fixing to have a baby. So she's going to go on maternity leave soon. This is the GNL. We did a tour of the GNL. And the last slide that I always leave with my mentoring slides is this slide. So what my mom thinks I do, what my friends think I do, what society thinks I do, what my boss thinks I do, what I think I do, but what we all really do, right, is juggle all kinds of stuff. So if you have any questions.
Any questions?
I have a technical question, kind of what you presented towards the end on the HIV PBMCs. So are these from patients that are elite super controllers? I might have missed that.
The PBMCs that we're collecting right now,
Yes.
they're from healthy donors. So we get Buffy packs from a blood bank. So none of them are HIV infected right now. We're just looking at whether cocaine or fentanyl will cause CD4 differentiation within healthy donors first.
Okay, thank you.
So I know you said you're not a big fan of immunology. Have you looked at any of the microglial response?
Yeah. So my research scientist, Shanita, one of the things that she's actually doing right now before she goes on maternity leave is she's submitting an R21 on microglia and autophagy during cocaine use in HIV. She also has looked at microglia immune responses with SARS-CoV-2. So we have dabbled a little bit in microglia. The problem is that we like to do a lot of our in vitro work with primary human cells, and microglia are relatively hard to get, to isolate from human tissue unless it's fetal, and we don't work with fetal anymore. And so she's been using immortalized microglia. So I'm kind of hesitant about the results that we see from that, but we've dabbled a little bit in it.
Thank you.
Any other questions?
Easier question, I promise. How do you this is very interdisciplinary research. How do you decide which center to submit your
My
proposal
grants
to?
too.
Yeah.
Yeah. So a lot of it has to do, I mentioned that I like to submit grants to RFAs because you have a higher probability of getting funding. So I actually scan the institutes that my research is relevant to. Usually it's NIAID, NIDA, NIA, NINDS, or NIMH. I look at their RFAs first and I see if my research will fit any of those calls for grants. And then that's my first level, right, of submission and that'll help me decide which institute would be primary. If I don't see anything and I have a grant that I think is ready to be funded, right now, most of my grants have been going to NIDA because it's cocaine or fentanyl use, but my Alzheimer's grants all go to NIA because it's an Alzheimer, or it's actually National Institute of Aging, but they did put an RFA for SARS-CoV-2 and Alzheimer's disease progression. So I think it's really, you have to be really careful when you do that. And when you submit a grant, you can kind of choose 3 institutes that you'd like your grant to go to. And when you're looking at program announcements or RFAs, it shows which institutes are funding them. And more often than not, there's multiple institutes that fund grant mechanisms. So it's not that hard to do it. It just depends on what the grant is on, really.
Hello, hi, great talk. Have you ever looked at the epigenetic changes in any of the inflammation processes or?
No, we haven't. I feel like I'm already kind of very expansive. If I had a trainee who was interested in that, I would be more than happy for them to move forward with that. That's also one of my things. If one of the individuals in my lab is interested in something, I allow them to explore it. So Shanita is submitting an R21 for autophagy and microglia, but that's not really my area of expertise. But her goal is to eventually open her own lab. So this is part of her training to submit this R21. So I personally am not interested in epigenetics, but if somebody else that is in my lab was, we would look at something like that.
Thank you.
Two questions also about grants, since you talked a lot about grants today.
Yeah.
So I think the first question is, how did you go from applying for fellowships like F31, F32 to applying to the R01? And what kind of learning process was that? Like, who did you learn it from? And like, what was the curve on that? Yeah.
So during my pre-doctoral work, actually my grant writing class was exceptional when I was a pre-doc. And when we did that, the process of grant writing was to develop an F31. So that was very beneficial for us because then we could turn it around and submit it to the Institute after it had already gone through review from several PIs, right, at the end, at the pre-doc level. And so that was the reason that I submitted my F31 the first time. And it was funded, thankfully. And for the F32, when I came on it with my T32 at UTMB, my goal was to submit an F32. And typically when you hear, when an institute funds an F31, they're investing in you. So they want to continue investing in you. So if they funded your F31 and you submit an F32, it's a natural progression in their investment. So my F31, my T32, my F32, my R01 are all from NIDA. And they look at me as an investment. They've given me money, they've seen me produce, and they want to continue funding you because it shows a solid track record. So that's kind of my advice to everybody to submit grants. They don't have to be to NIH. There's all kinds of institutes to submit, but it looks really great, especially if you want to stay in academia. So submit.
Well, what's your really big difference between applying to fellowships versus applying to the bigger grants?
Like the R01 year?
Right, the R56, R01 and whatnot, yeah.
So,
And how did you kind of deal with that gap?
right. A lot of the programs that I had showed that I was involved with, like NIH Future Leaders, NINDS Brains, the NIDA Diversity Fellows Network, all of these were specific to grant writing. And so every program that I've applied to through NIH has provided me with grant writing guidance. And so that's really led me to submit grants. My goal is to submit 3 grants, either new or resubmissions every year. So every cycle I like to submit a grant. It's not easy and it's frustrating, but what I actually really like about grant writing is that it's a persuasive document. It's not so much scientific, right? So I teach grant writing at UTMB for our expat students. And one of the first things we do is we give them the analogy that NIH is a bank and you're asking for a loan. So you have to persuade them to give you the money. And so it's very different from writing a manuscript or anything like that. And you get to be more creative with grant writing, which is what I really like. So, yeah.
Okay, from the chat. What would you advise to a graduate student that does infectious disease research and is considering to transition to neuroinfectious diseases after graduation?
So I think that identifying a postdoc position that's within a neuroinfectious disease laboratory is going to be really important. But if you are really interested in going to an infectious disease laboratory that doesn't do neurovirology research, be prepared to pitch something to that PI that you'd like to work with, where you are persuading them to adjust their research into a different direction and use the literature to support that investment that they're going to make in you looking into neurovirology.
Any last questions? Okay. Well, thank you very much for your talk
Thank
and
you.
covering all of the specifics of grants. So.